Whether you're exploring disease mechanisms, identifying the right targets, optimizing lead compounds, or assessing ADME or tox risks, the volume and fragmentation of data can slow you down. In this video, you'll discover how to extract insights faster and make better-informed decisions using CAS BioFinder. We'll begin by exploring how CAS BioFinder can help biologists understand more about the disease they're investigating, so they can properly identify potential targets. When investigating a disease, the first step is to assess the current disease biology and therapeutic area. CAS BioFinder brings everything into one connected view, so biologists can quickly understand the relevant context and move forward with confidence. Let's use osteoarthritis as an example. On the osteoarthritis disease overview page, you can access a concise summary that includes a clinical description and a full list of synonyms to ensure alignment and consistency when connecting results with public data or collaborators. You can also view the protein page for osteoarthritis to assist in identifying potential targets that will affect it. Let's look at how CAS BioFinder can help biologists identify the right biomarkers to understand disease mechanisms. Biomarkers can help you connect molecular signals to disease mechanisms, whether it's tracking progression, evaluating treatment response, or identifying points of intervention. That's why access to reliable, well-contextualized biomarker data can be the difference between advancing tomorrow's therapeutics or pursuing costly dead ends. In the Biomarker tab, you can view a wide range of curated data. For example, by understanding how the biomarker interleukin-11 relates to the genetic basis of hip osteoarthritis, you can begin to understand how this disease plays out in the body and how it can be treated without a hip replacement. Filter by category, such as diagnostic, prognostic, or pharmacodynamic biomarkers, to shift focus depending on research goals. Pharmacodynamic markers are particularly useful for understanding disease mechanisms, as they reveal how the body responds to treatment based on measurable changes tied to specific drugs and doses. You can also select specific biomarkers to narrow the list down further and review detailed assay results. This can make it easier to see how biological changes relate to the disease and where the biology might be most responsive to targeting. CAS BioFinder makes it easy for biologists to quickly identify which protein targets are most relevant for their disease of interest. In the Pharmacology tab, you can review well-known as well as less studied protein targets associated with the disease of interest. For example, selecting prostaglandin GH synthase 2 provides access to hundreds of target compound interactions to consider. Too much data? Too little time? Use advanced filtering to cut through the noise and bring focus to what matters. With a single click, you can review assay conditions and bioactivity results further. These results are supported by trusted literature references, so you can advance your drug discovery workflow without wasting time second-guessing data. CAS BioFinder organizes structural, functional, and pharmacological data all together in one place to help biologists better understand how target proteins function in the body and interact with ligands. We'll use prostaglandin GH synthase 2 as an example. You can find 3D structures derived from X-ray, cryo-EM, or NMR studies directly sourced from PDB. See how the ligand interacts with specific amino acid residues at the binding site to support structure-based analysis of affinity and selectivity. You can explore compounds known to interact with their target protein and review associated bioassay results to better understand target compound interactions. Review known or predicted metabolites to compare activity across chemical series and prioritize compounds for further study. See how the target fits into broader signaling networks. You can trace upstream regulators and downstream phosphorylation events. This can help you to better anticipate functional impacts of target modulation and evaluate potential downstream effects. Repurposing existing drugs can accelerate development timelines and reduce risk. CAS BioFinder provides access to the detailed, connected drug information discovery teams need to find new indications and opportunities for existing drugs. AI drug intelligence summarization provides an overview of approved, investigational, and withdrawn drugs, so you can quickly get up to speed with the current therapeutic landscape. Let's look at piroxicam as an example. Review the CAS Drug Intelligence Summary with key information, such as development history and approved indications. Piroxicam is indicated for osteoarthritis and rheumatoid arthritis. This high-level summary also provides an overview of piroxicam's primary targets. But you can also view other compound-target interactions. You can assess piroxicam's bioactivity on various protein targets to get a clear view of compound behavior in disease-relevant models. By filtering for specific cell lines, teams can compare IC50 values and assay results from earlier studies, which could be especially helpful for identifying niche indications or refining model selection. For example, you can evaluate piroxicam's antimicrobial or antiviral potential. Select specific bacteria and viruses in the lateral filtering panel to review organism-relevant bioactivity data. To further support effects repurposing an existing drug, CAS BioFinder provides easy access to curated toxicology data and known and predicted metabolites. This enables you to quickly evaluate compound safety and explore mechanisms of action in new disease contexts. Explore similar ligands and scaffold variations to identify compounds with different biological profiles or target selectivity. CAS BioFinder empowers discovery teams with predictive analytics, so they're not limited to only what's already been published. Now you can anticipate off-target interactions and uncover novel target associations for a head start on developing new hypotheses for drug repurposing, even when the literature is missing. CAS BioFinder helps medicinal chemists identify ligands with high research potential, so they can focus their resources on drug candidates that are most likely to be best-in-class therapeutics. To evaluate ligand activity, chemists can start by viewing all compounds tested against a target protein. For example, we'll view all compounds tested against prostaglandin GH synthase 2 in the context of osteoarthritis. In the Pharmacology tab, you can review the number of measurements per compound and also narrow the list down to only the most relevant compounds with advanced filtering capabilities. Each result includes IC50 values, assay types, and links to source literature, so you can assess how potent each inhibitor is, how the data was generated, and in what biological context. CAS BioFinder provides a built-in property-based filter set to help you focus on the most promising ligands. Alternatively, they can fine-tune parameters to better assess drug likeness and filter out compounds that may face issues with solubility, permeability, or oral bioavailability. Before advancing a ligand, it's essential to understand where else it might be active. Going beyond just your primary target, CAS BioFinder enables you to explore both known and predicted targets, so you can flag potential off-target effects, metabolic liabilities, or early safety concerns. For example, piroxicam shows associations with cytochrome P450 enzymes and carbonic anhydrases. CAS BioFinder's predictive models can highlight activity even when no measured activity is available. Once you've identified promising ligands, you can explore the surrounding chemical space to identify analogs or spot structure-activity trends. Map related compounds in 2D or 3D graphs like log-P versus molecular weight. You can also navigate similar scaffolds to see how structural changes affect properties and potency as well. In one connected workflow, CAS BioFinder can help you identify, evaluate, and optimize the most promising ligands by integrating target data, predictive modeling, and chemical insights. CAS BioFinder was designed with lead optimization in mind. Now, medicinal chemists have the tools to confidently refine candidates so they can maximize potency and minimize off-target risk to avoid late-stage surprises. Explore how small structural changes may impact bioactivity by using matched molecular pairs analysis. Compare analogs that differ by a single substituent and see how those changes affect potency across assays. This key feature can help identify activity cliffs, so you can select the best ligand to explore as a drug candidate. You can also analyze R-group substitutions at specific scaffold positions. CAS BioFinder highlights how different substituents impact activity at each site on the molecule to give you a clear view of structure-activity relationships and help prioritize which modifications to explore next. Once the top ligand candidates have been identified, you can use CAS BioFinder's built-in predictive analytics to screen them across thousands of targets. This will help estimate how available the ligands are for binding and where they might cause off-target effects. Choose from broad pharmacology panels, preset safety panels, or build a custom panel tailored to the therapeutic area of interest. You can also explore similar ligands that have advanced to the clinic. You can review predictive toxicity profiles to anticipate possible setbacks and avoid costly safety pitfalls. CAS BioFinder can help you streamline lead optimization, refine candidates with confidence, and reduce late-stage risk by revealing how structural changes impact candidate activity, safety, and selectivity. Optimizing a novel drug candidate means predicting how it might behave and if it can be made. CAS BioFinder helps medicinal chemists evaluate potency, safety, and synthetic feasibility all in one workflow. You can begin by securely uploading your leads to screen them against CAS BioFinder's pharmacology panels. This allows you to uncover predicted activity across known and unmeasured targets, so you can explore new mechanisms and flag off-target risks, even before any lab data is available. Once you've screened your novel molecule, you can explore known ligands with similar structures. CAS BioFinder will surface salts, prodrugs, and analogs that you can filter by key drug likeness features, like molecular weight, log-P, and polar surface area to compare. Next, you can then use CAS BioFinder's R-group substitution analysis to explore how different substituents affect potency and safety. You can view specific positions on a scaffold and compare real-world bioactivity data across matched molecular pairs to identify substitutions that improve activity, reduce risk, and remain synthetically accessible. Once you have optimized your design, you can jump into CAS SciFinder directly from the molecule page to generate a retrosynthesis plan. CAS SciFinder provides access to published reactions, route suggestions, and starting materials to help you move quickly towards synthesis, scale-up, and in vivo testing. CAS BioFinder unites predictive pharmacology, safety screening, and synthetic planning to help you design, evaluate, and advance safer, more feasible drug candidates with confidence. CAS BioFinder is your partner in uncovering the connections, data, and insights you need for breakthrough discovery. Whether you're a discovery biologist or medicinal chemist, CAS BioFinder provides you with a single, trusted platform to help you move with speed and confidence. Connect with us to learn more, request a demo, or start your own journey with CAS BioFinder.